Hi everyone, today I'll be going over the Biology Chapter 7 Photosynthesis of the course revision sheet for grade 12 final exam. Firstly, we have what is photosynthesis? It's the process in which plants synthesize carbohydrates from carbon dioxide and water. Over here you have the equation that demonstrates this reaction. It consists of two reactions, the light-dependent reaction and the light-independent reaction. It occurs in the chloroplast of plant cells. We'll go more in depth for the reactions. So first up we have light-dependent reaction, the non-cyclic photophosphorylation. Over here we have a chloroplast and then we're zooming in on the thylakoid membrane. This is where the light-dependent reactions take place. Although it's mainly on the thylakoid membrane, the thylakoid itself plays an important role, like the thylakoid space inside here and then on it, like the membrane. So when they ask you where does the light dependent reaction take place, just say thylakoid because it contains everything. So firstly we're going to start with water. What happens to water? Light energy hits the water, so like the water will absorb this light energy and it will split into three components. It's split to hydrogen ions, O2, which is oxygen, and electrons. The hydrogen ions play an important role in the electrochemical gradient in the thylakoid lumen, or the thylakoid space, which will be later used for ATP synthesis. The O2 is released as waste, and the electrons transported to photosystem 2. So when it's in photosystem 2, light energy excites this electron. So now it's excited, so basically unstable. It's going to move from this electron to from this photosystem 2 to the photosystem 1. It goes through here. And as it goes through, it enters, like it passes by the electron transport chain, which contains these type of proteins, cytochromes and PQ. And basically the role is to, like, as the electron passes through, energy is formed over here. So since you have energy, the hydrogen ions will go from the stroma to the thylakoid lumen. In the stroma there's a low concentration of hydrogen ions, but we're trying to build a concentration gradient from the higher being in the thylakoid space. So it's going to go from active transport from the stroma to the thylakoid space. And then as it reaches photosystem 1, it's going to be excited again, and it's going to pass through from photosystem 2 to NADP reductase. This is a place where NADP plus gets reduced to NADPH. And then since we build up all the hydrogen ions over here, so the concentration is really high, it's going to go through this thing called ATP synthase, which as it goes through, ADP and inorganic phosphate will combine and form ATP. The energy comes from the hydrogen ions flowing down the concentration gradient, and this energy is kinetic energy because it's like flowing. So this process is called chemiosmosis. And then our end products are ATP and reduced NADP, which is the same as NADPH. Let's have a closer look to the photosystems. Over here we have photosystem 2, and you don't have to memorize this diagram. I'm just showing it for a better understanding. So over here we have water, which is, as we said before, split. Oh, and this reaction is called photolysis. It's when water absorbs this light energy and splits to electrons, oxygen, and hydrogen ions, which are also called protons. So the electrons coming from water to photosystem 2, and it goes to the primary pigment. Over here it says primary electron acceptor, but I think it's the same as the primary pigments. You'll see it in the course revision sheet. And then light excites this electron, and the electron moves to photosystem 1. The electron comes from photosystem 2. So from photosystem of the replacement electrons for photosystem 2 is water, the photolysis of water, and the replacement electrons for photosystem 1 is the electron from photosystem 2. So as it's here, it goes to the reaction center, which is the primary, wait, never mind, not reaction center. It goes to the primary pigment, like I think this is the primary pigment again, which is the primary electron acceptor. Now over here we have a light-dependent reaction, the cyclic photophosphorylation. So the difference between the cyclic and non-cyclic is that the cyclic only involves PS2, which is PS1, sorry, which is only photosystem 1. The excited electrons go through the electron transport chain, which is a series of electron acceptors and the proteins like cytochromes and PQ, and then they return back to PS1. As it goes through the electron transfer chain, energy is released to generate ATP by chemiosmosis. So remember how we said that the hydrogen ions are pumped against their concentration gradient, and like as the electrons move by, the energy helps this active transport. So as the hydrogen ions move by, it's like an ATP synthase, but instead of that we're in the cytochrome and the PQ, I think. So since hydrogen ions move by, you're going to have energy, and this energy will contribute to chemiosmosis, which is the production of ATP by the hydrogen ions concentration. Hydrogen ions like flowing through the protein. In cyclic photophosphorylation, our end products are ATP only. So now we have light-independent reaction, or another name is called Calvin cycle. So there's 3 main steps for this Calvin cycle: carbon dioxide fixation, carbon dioxide reduction, and ribulose biphosphate regeneration. These are the 3 important steps that will come in the exam. So let's talk about carbon dioxide fixation. 1 carbon dioxide reacts with a 5-carbon sugar, which is called ribulose biphosphate, or RUBP in short, to produce 2 glycerate 3-phosphate molecules. We use the enzyme RUBP carboxylase to catalyze the reaction. Next we have carbon dioxide reduction. The glycerate 3-phosphate molecules are reduced by NADPH and ATP to produce triose phosphate, also known as glyceraldehyde 3-phosphate. So where do these NADPH and ATP come from? They come from the light-dependent reactions of the NADPH comes from the light-dependent reaction, the non-cyclic one, and ATP comes from both of the cyclic and non-cyclic reaction. Lastly, we have RUBP regeneration. Though some triose phosphates leave the cycle to produce glucose, amino, or lipids, most are in fact used to regenerate RUBP, so the cycle can continue and so on. Here's a summary for the photosynthesis. We have light-dependent reactions which take place in the grana or thylakoids of the chloroplast, and light-independent reaction which take place in the stroma of the chloroplast. So in the light-dependent, light energy is converted to ATP and reduced NADP or NADPH. It can be a cyclic or non-cyclic, and oxygen gas is released as a waste product. For light-independent reaction, it consists of 3 main stages as we discussed before: carbon dioxide fixation, carbon dioxide reduction, and RBP regeneration. These steps make up the Calvin cycle, and no light or light energy is used because although light-independent reactions need the NADPH and the ATP, they all come from the light-dependent reactions. Thank you for watching, and good luck in your exams.